Epilepsy is a disease that causes unpredictable and often violent seizures, and it affects 3 million people in the United States. Many forms of epilepsy impact an area of the brain called the hippocampus, which plays a role in learning and spatial awareness. There are treatments that vary in effectiveness, but no known cures. The best way to create better treatments and potential cures is to understand the disease at the most basic level, the level of the neuron. Neurons are the cells that make up the brain, and they communicate to each other through electrical messages. During epilepsy, the neurons randomly fire, which overloads the other neurons with information and causes a seizure. We know that epilepsy causes physiological changes to the neurons it affects, meaning that the messages the neurons send and the way they send them significantly change. What is not known is what causes these changes. Is it because of structural changes to the neuron or to changes in the proteins in the cell? Through my research, I hope to answer the question of whether epilepsy in the hippocampus causes significant structural change to the neurons it affects. I use a technique called DAB that results in staining a single neuron in a brain slice from a rat hippocampus, which allows me to build a computer model of the neuron through a technique called reconstruction. From here, I can use computer software to measure characteristics such as amount of branching, surface area, and length to quantify the structure of a neuron, and then compare the differences between healthy and epileptic neurons. Understanding the specific things that happen to neurons during epilepsy is an important step to better understanding the disease, which will allow us to create treatments and cures that target the root cause rather than just the symptoms.